Hi, my name is Eric and welcome to another video in cell biology. Today we're going over the plasma membrane. The plasma membrane consists of membrane proteins, phospholipids, the phospholipid bilayer. Membrane proteins allow for selective permeability with integral and peripheral proteins. Integral and peripheral proteins, we have different kinds here: ion channels, enzyme proteins, proteoglycans, receptor proteins, transporter proteins, anchor proteins. Ion channels allow for passive transport of ions. Active transport allow for transfer of ATP, making other molecules like ATP, for example, ATP synthase. Proteoglycans are proteins bound to sugar, and this allows for membrane protection. There are receptor proteins on the outer membrane, surface of the membrane, and also on the inner surface near the cytosol. These allow for cell growth, so any response from the extracellular environment will allow for a chain reaction to occur to allow for the cell to start dividing. There's an electrochemical gradient that's regulated here for the transport proteins, and those proteins are actually sort of like pumping hydrogen ions in the electron transport system. And the link of actin filaments, which makes up sort of the skeleton of the cell, is anchor proteins like integrins. If we go here, the phospholipid bilayer is consisted of amphipathic molecules that are nonpolar and polar molecules. The phospholipid head is hydrophilic, the phospholipid tail is hydrophobic, and according to two different theories, several decades ago, Davison and Danieli came up with the sandwich model, which their theory was that the cell membrane is consisted of a lipid bilayer, and or some sort of lipid layer, and that was actually proven much later on, probably a couple decades later, that this was true using electron microscopy. There's also another model, which is the fluid mosaic model, that was first introduced by Singer and Nicholson, and they had a theory that the cell membrane divided the intercellular organelles and materials from the outside and allowed for selective diffusion, which came up to be sort of a selective permeability and what we know as the cell today and how it's regulated on the surface. Selectively permeability, this just allows for any ions, molecules, proteins to go from one side of the cell to another, and so that's highly regulated by the proteins and obviously the vesicles during endo and exocytosis. The two components that make up the membrane, which are the proteins and phospholipids, also regulate a concentration of molecules and water on both sides. Anything that's hypertonic is a high concentration of molecules on the outside of the cell, or inside of the cell, sorry. And the hypotonic is the concentration is very high on the outside, so the cell essentially gets larger in a hypotonic solution versus hypertonic, which makes the cell smaller. Concentration gradient is also controlled by this phenomenon called osmosis, which explains that anything that has molecules coming from one side to another is regulated by the flow of one molecule, a group of molecules to another. So if there are more on the outside of the cell, water is going to actually dissipate from this location to the inside. So For example, if there's less here, the water will flow into here, and some of the molecules will flow out here to kind of stabilize the environment, which brings us to isotonic, which there's a stable concentration of a solvent in a solution on both sides of the cell membrane. And then also, oh, I forgot these up here. Glycolipids are the carbohydrates that are bound to phospholipids, and they're responsible for cellular responses and cellular communication. And that concludes my presentation of the plasma membrane. Thank you for watching this video, and have a nice day.